Welcome to REACTOR-X, the lab where science truly comes alive. Today we're guiding in what have been Autolysis pod. Bones aren't just rigid structures. They are composed of collagen protein matrix, calcium phosphate minerals, living bone cells called osteocytes. Now, let's simulate autolysis using a safe lab model. When these enzyme solutions interact with the protein model, they begin to break down the structure. Watch closely. Autolysis is a biochemical cascade. When a cell loses energy, pH levels drop, lysosomal and control system failing. Bones are more resistant because of their high mineral content. That's why autolysis affects surrounding soft tissues first. Here in this time-lapse simulation, you can see the protein matrix gradually breaking down under enzyme exposure. Autolysis and tissue preservation, quality, and the cellular synthesis. Today, we explored autolysis and enzyme-driven cellular breakdown in a safe simulation. Next, explosive reactions.
Welcome to REACTOR-X, the lab where science comes alive and curiosity meets reality. Today, our focus is on one of the most fascinating processes in biology: autolysis, the self-destruction of cells after life ends. Earlier, Dr. Arman Hale demonstrated this entire process for us in the lab, using safe, visually engaging models, so we could fully understand how tissues and cells begin to break down at the molecular level. Dr. Hale began by showing the internal structure of a typical cell. Using a detailed holographic display, he pointed out the lysosomes, tiny organelles packed with enzymes that have the power to break down proteins, lipids, and other cellular structures. While the organism is alive, these enzymes are tightly regulated, held safely within membranes, and controlled by the energy of the cell. He explained that as long as the cell produces ATP and maintains oxygen flow, these enzymes remain inactive. Think of them as tiny demolition crews, he said, waiting for the right signal to start their work. But if the cell loses energy, these crews are unleashed. Next, Dr. Hale turned our attention to tissues and bones. He used a protein-based tissue model to represent soft tissue and a mineralized model to show bone structure. He explained how bones contain collagen fibers for flexibility, calcium phosphate for rigidity, and osteocytes as living cells embedded inside. The demonstration began with soft tissue, showing how enzyme solutions interacted with the protein model. Slowly, the matrix started to soften, small fragments broke away, and bubbling appeared as a simulation of enzymatic activity. This is exactly how autolysis begins in a living organism, Dr. Hale said. The enzymes, once unrestrained, gradually dismantle the cell from within. He then highlighted the differences between soft tissue and bone. While soft tissue succumbs quickly, bone resists breakdown due to its mineral content. But he also showed how, over time, even bone weakens once surrounding tissues are gone and environmental factors contribute to decomposition. Using color-coded holograms, he illustrated the stepwise degradation: membranes losing integrity, proteins unraveling, lipids dissolving, and eventually, the structural collapse of tissue. The visuals made it clear that autolysis is not random. It is a precisely orchestrated chemical and enzymatic process. After showing the lab simulation, Dr. Hale explained the process of autolysis in living organisms after death. He pointed out that immediately after life ends, cells lose their ability to produce energy. Without ATP, membrane pumps fail, causing the cell to become permeable. Enzymes, previously contained, flood the cytoplasm and begin digesting the cell's own proteins, lipids, and organelles. pH levels drop, creating an acidic environment that accelerates enzymatic activity. He emphasized that this is the very first reaction in postmortem decomposition, the internal self-digestion of cells that starts almost immediately after death. The narrator continues, "Every living organism, from the simplest microbe to complex mammals, undergoes this process in remarkably similar ways. Soft tissues, muscles, liver, and internal organs, begin to break down first. Proteins denature, lipids dissolve, and structural molecules fragment. Lysosomal enzymes, now active, accelerate the destruction, releasing metabolites and chemical byproducts into the surrounding environment. These byproducts not only soften the tissue but also prepare it for further decomposition by bacteria and other microbes. Bones and other mineralized structures resist initially, but over time, chemical changes and enzymatic activity gradually weaken them. Calcium phosphate crystals in bones slowly dissolve in response to environmental factors, and collagen fibers break down under enzymatic attack. Autolysis is a cascade, a chain reaction in which failure of one system triggers the next. Membrane permeability changes lead to enzyme release, which lowers pH, which activates more enzymes, eventually collapsing cellular architecture entirely. Dr. Hale also explained that autolysis is the reason why decomposition begins internally before external factors like microbes play a major role. It is the organism's own chemistry that initiates the process. Metabolic byproducts, acidic conditions, and internal enzyme activity combine to make tissues soft, prepare them for bacterial colonization, and ensure that nutrients are recycled back into the ecosystem. He highlighted that even after the body stops functioning, chemistry continues, a silent, invisible, yet powerful set of reactions that manage the transition from life to decomposition. The narrator emphasizes that autolysis is universal. Every living organism, after death, undergoes these chemical reactions in a remarkably consistent sequence. Membranes rupture, pH drops, enzymes activate, proteins and lipids break down, metabolites spread, and tissues lose integrity. This sequence ensures that soft tissue is recycled efficiently, while bones and other mineralized structures degrade more slowly. Environmental conditions, temperature, moisture, and oxygen, affect the speed but not the underlying sequence. Finally, Dr. Hale walked us through the broader picture. Autolysis is not just decomposition. It is nature's way of recycling life's components. Proteins break into amino acids, lipids into fatty acids, carbohydrates into simple sugars, and minerals from bones return to the soil. These reactions are invisible yet fundamental, powering ecosystems by returning vital elements like nitrogen, carbon, and phosphorus to the environment. The scientists' demonstration made it clear that the process is highly organized, predictable, and chemically fascinating. As the video closes, the narrator reflects, through Dr. Arman Hale's demonstration, we saw step by step how life ends at the molecular level. We witnessed the first signs of cell self-destruction, the chemical cascade that begins immediately after death, the sequential breakdown of tissues, and the eventual weakening of bones. Autolysis is a powerful reminder that even after life ceases, biological and chemical laws continue to govern the transformation of matter. Here in REACTOR-X, these processes are visualized, studied, and understood safely, providing an incredible insight into the hidden chemistry of life and death. If you enjoyed this exploration, subscribe to REACTOR-X, where every experiment brings science to life, and every reaction reveals the invisible, essential processes that sustain and recycle our world. Next time, we will delve into glowing chemical reactions, explosive laboratory simulations, and molecular transformations that will once again show the beauty and power of chemistry.